Okay, so this part of the video, we're going to look at synaptic transmission in general, and then there'll be another video with synaptic transmission for muscle also. It's just slight difference in what's going to happen. Mostly the slight difference has to do with the neurotransmitters that are going to be released. Okay, so let's draw a couple things on here. So I gave you guys this picture in case you can't draw the synapse, then you can just fill it in. And then we're going to go through the processes like 1, 2, 3, 4 steps, however many steps I make. But let's draw a couple of things first, or for you guys, maybe label a couple of things. So here's our axon terminal. The axon terminal region leads to the bulb, which is the synapse. Remember, we drew this already on our generic cell, okay? On our, our generic neuron, or our typical neuron. This is going to be my receiving cell or my target cell. The receiving cell can be a neuron. It could be a muscle. What type of muscle? Skeletal muscle, cardiac muscle, smooth muscle. Or it could be a gland. Anything that the neuron's going to send a message to, basically. That's what this part could be, okay? So between the synapse and the receiving cell is an area that is going to be called the synaptic cleft. This is basically just space between the two cell membranes, and this will be filled with interstitial fluid. Okay, remember, interstitial fluid is the fluid that goes around our cells, it bathes all our cells. Inside of the synapse, we're going to have little membrane sacs, and these membrane sacs are going to hold neurotransmitters. I'm going to make neurotransmitters a different color than the colors we used before. So these are neurotransmitters. So we have our synaptic vesicles with our neurotransmitters. Okay, there are different types of neurotransmitters. Those neurotransmitters are going to either be excitatory or inhibitory, meaning they either cause an action potential or they prevent action potentials. As I said in class, the only one you need to know is acetylcholine, and that's for muscle contraction. So you can kind of put that right here. So muscle neurotransmitter is going to be acetylcholine. Its abbreviation is big ACH, okay? The other thing that we're going to need is we're going to need receptors and channels here on our receiving cell. And I'm going to make those in red because what I want you guys to remember is that these are sodium channels. Because we're only going to deal with excitatory neurotransmitters. And then I usually put like a little kind of half cup or a little cup here on the side. And that little cup is going to be for my neurotransmitter to bind to. So the receptor area on the channel is going to be specific for our neurotransmitter, okay? So we kind of drew everything. I think we drew everything that we need. Now we're going to do the process and the steps. I might not write out every step. So you might want to like do a 1, 2, 3, 4 and write this out as you follow along. I'm just not going to write it out because it takes longer. I might write it out. We'll see. Okay, we're going to go backwards in our stuff that we drew, and we're going to go back to depolarization and action potential because I want you to kind of connect all of these things together. So we want to remember sodium has entered into our cell. We become negative 55 millivolts. And when we become negative 55 millivolts, we open the next sodium voltage gated channel all the way along our axon until we create an action potential. Okay, so we're traveling along our axon. And what we're going to see is that as we travel along our axon, like that, we'll have this action potential coming along our axon terminal. So this will be our action potential, okay? And I actually forgot to draw in the most important thing is, well, one of the most important things is that we're going to have calcium now, and we're going to have calcium voltage gated channels, okay? So in our synapse, instead of having sodium voltage gated channels or potassium voltage gated channels, it's the calcium voltage gated channels that are now going to be more important. And what we're going to see is that there's going to be more calcium sitting outside of our cell at this time than sitting inside of our cell. Okay. So as the action potential comes down the axon terminal, we're going to remember this is a positive charge. As that positive charge reaches our voltage gated channel, it's going to pop open our voltage gated channels. And as we pop open our voltage gated channel here for calcium, we're going to think about the fact that calcium is much higher on the outside than it is on the inside, right? Well, so we're going to go from our high concentration to our low concentration. So calcium is going to enter into the cell. So we'll have much more calcium here, okay? What happens is the calcium will actually bind to the synaptic vesicle, and it causes the synaptic vesicle to start to move, and the synaptic vesicle will fuse with the cell membrane of the synapse. And when it does this, it's basically going to open and release the synaptic vesicle into the synaptic cleft. So what that's going to cause is all of our neurotransmitters to basically be released into the synaptic cleft, right like that. Okay. Those neurotransmitters will then come and find their correct receptor, and they'll bind to the receptor that is on our sodium channel here, which I should probably label what that is. This is a sodium channel. It's not a voltage gated channel. This is given the term a ligand gated channel. I don't like to use that word for your class too much because it's a little confusing. So I just tell you it's a sodium channel, okay? But it's not voltage gated. So it opens because of the neurotransmitter that binds to it. So when the neurotransmitter binds to the sodium voltage gated channel, each one of these is going to open. And I want you to remember, this is a receiving cell, and that receiving cell has to also be electrically charged. So that means that it's negative on the inside and positive on the outside. And it's positive on the outside because we have a whole bunch of sodium sitting outside of our cell, like we normally do. So as our neurotransmitter binds and we open these channels, then we're going to see that sodium is going to rush into our cell here, and it's going to cause our negatively charged cell inside to become positively charged, like that, on the inside. And this is going to result in creating an action potential that goes along our cell. So if this is a neuron, this is the stimulus that I need in order to open my first sodium voltage gated channel. So at this point, you think about like this is the dendrite, and here I'll have my sodium voltage gated channel along my axon. As soon as this becomes at least negative 55, this opens my first sodium voltage gated channel right here. And basically I start depolarization. Then I can open the next one, the next one, and the next one, and the next one. Okay. If this is a muscle, it's very similar. You have sodium channels that open, and this will cause another sodium voltage gated channel to open, and you'll have an action potential that you create along your muscle membrane or your muscle cell membrane. The only difference is for a muscle, the action potential can go in all directions. For a neuron, it can only go in one direction. So as soon as you create your action potential for a neuron, it goes down the axon in one direction. But for a muscle, it's going to spread along the entire cell and go in all directions. Okay. So I'm hoping that this also helped you kind of visualize a little bit synaptic transmission, but I will also post a video that I have from a publisher as well that's more of an animated 3-dimensional video to help you like visualize what these channels and ions moving actually is going to look like. So after all of these videos, you should understand how to draw a typical neuron, what are the functions of each part of the typical neuron. You should understand the complicated process of neuron physiology. What is the process of resting membrane potential, depolarization, repolarization, hyperpolarization, and synaptic transmission? And you should hopefully have filled out your graph completely here, which I forgot almost to finish synaptic transmission. Good thing I went back here. So for synaptic transmission, I want you to pause again and try to fill out this chart really quickly. Okay, hopefully you came back after you paused. I hope you paused. We're going to see that sodium is still going to be high outside of the cell, but for synaptic transmission, you might want to just negate that because I don't want you to be confused about what happens to sodium. Sodium and potassium are not important here. What is important is that calcium is going to be high outside of the cell, and then it becomes high inside of the cell. So we really want to think about it's going to be high inside of the cell because we open calcium voltage gated channels, okay? This part of the chart I kind of threw in after I created it. It's not the best part of the chart. You want to make sure you can draw all that out. Make sure you understand that chart. That chart is very helpful, I think, in the whole process of where all your ions are, where your ions move to, okay? And then post your questions if you have more questions about all of these processes or come to office hours.